So what happens when you deploy an immunotherapy that completely relies on CD8 positive T cells to kill a tumor, but you deploy it against a cancer that has already neutralized those exact cells?
Well, by all conventional oncology logic, it should just fail.
Yeah.
I mean, it really shouldn't even make a dent.
Right. But in this massive new study looking at pancreatic cancer, a new targeted combination didn't just work. It completely eliminated the tumors.
Yeah, it's truly a paradigm-shifting discovery. We are basically looking at a fundamental rewrite of how we understand immune activation.
Especially against highly evasive malignancies, right? So welcome to another deep dive. Today, our mission is to crack the armor of one of the most stubborn and deadly cancers out there.
Right, pancreatic ductal adenocarcinoma.
For PDA.
Exactly, PDAC. And we're using a fascinating 2026 preview article from the journal Cancer Cell by researchers Cruz, Leonhardt, and Butcher as our map today.
And they're synthesizing this groundbreaking new research by Qureshi and colleagues. Yeah, so for you listening at home, we're going to break down exactly why traditional immunotherapy fails so spectacularly against PDA and how a brand new triple therapy bypasses those failures entirely.
And we really want to get into the specific mechanisms today. This isn't just an observational win.
Right, they actually tracked the cellular relay race step by step to prove how this whole new immune axis functions. Okay, let's unpack this.
I'll do it.
We need to start with the baseline of why PDA is such a notoriously hostile environment. I mean, if you follow oncology, you know that the immunosuppressive tumor microenvironment in PDA is, well, it's essentially a fortress.
That's a great analogy. It is highly fibrotic, it's highly hypoxic, and it actively recruits immunosuppressive cells right to its borders.
So actively defending itself against our immune system.
Exactly. And to understand the breakthrough, we first have to look at why our standard, modern miracles like immune checkpoint blockade therapies, or ICB, why they fail here.
Right, the ICBs.
Yeah, when we talk about therapies utilizing anti-PD-1 and anti-CTLA-4, we are relying heavily on a very specific duo of immune cells.
I always picture them as the classic frontline infantry and their spotters.
I like that.
Right, so you have your cytotoxic CD8 positive T cells. They're the infantry, they do the actual killing.
Correct.
And then you have your type 1 conventional dendritic cells, or CDC1s, and those are the target spotters.
Yeah, the CDC1s acquire the tumor antigens, they present them, and essentially they point the CD8 T cells in the exact right direction.
And in cancers like melanoma or lung cancer, when you apply ICB to, take the molecular brakes off the immune system, this CD8 and CDC1 duo goes to work, right?
Absolutely. You get these durable, incredible clinical responses.
But in PDAC, the tumor microenvironment is incredibly efficient at shutting down that exact duo.
It is. It completely curtails the activity of both the CDC1s and those tumor-infiltrating CD8 T cells.
So the spotters are basically blindfolded, and the infantry's weapons are jammed.
That's exactly what's happening. So when you give a PDA patient standard dual ICB therapy, the tumor remains largely unaffected. It's totally insulated.
Wow. Okay, so if I'm tracking the biological math here, we have a scenario where the tumor has effectively locked the front door.
Right.
The standard heavy hitters, the CD8 T cells and the CDC1s, are completely neutralized, which means if you are going to attack this tumor, you can't just, try to force that jammed front door open.
No, you have to find a biological backdoor.
Let's pause right there for our first 5-minute checkpoint just to make sure you, the listener, are tracking all this before we get into the crazy new treatments.
Sounds good.
Okay, here's the recap. Bullet 1. PDAC is heavily armored against immune cells, basically acting like an immunosuppressive fortress.
Bullet 2. Traditional ICB therapy heavily relies on two specific cells, the CD8 positive T cells and the CDC1s.
And bullet 3. PDAC actively suppresses these specific cells, making standard immunotherapies practically useless.
Which perfectly sets the stage for what Qureshi and colleagues decided to test to find that backdoor.
Right. So let's get into their intervention. Because they utilized multiple murine models, mouse models of pancreatic cancer.
Yes. And crucially, they didn't just rely on subcutaneous models where you inject the tumor under the skin.
Which is easier, right?
It is, but it's not as accurate. They used orthotopic models, meaning they implanted the tumors directly into the pancreas.
So they could accurately replicate that highly immunosuppressive native microenvironment we just talked about, the real fortress.
Exactly. And their intervention to find this back door was what they called a triple therapy.
Okay, what's in the triple therapy?
Well, they took that standard systemic dual ICB, the anti-PD-1 and anti-CTLA-4, and they combined it with an intratumorally injected STING agonist.
Wait, a STING agonist? STING stands for stimulator of interferon genes, right?
That's right.
And if I remember correctly, STING acts like a massive localized fire alarm for the innate immune system.
That is the perfect way to describe it. So they are essentially giving these systemic checkpoint inhibitors into the bloodstream, but also dropping a molecular flare right inside the tumor fortress.
I love that visual. And the visual data in the source text, particularly figure 1, is just striking.
It really is night and day.
Yeah, in the cohorts receiving only the dual ICB therapy, you see rapid, completely uninhibited tumor progression.
The tumors just keep growing.
But in the cohorts receiving the triple therapy, the systemic ICB plus that local STING agonist, you see complete tumor elimination.
Highly effective, durable control of those PDAs tumors.
Okay, but here's where it gets really interesting. And I want to isolate the paradox here, because when you read the underlying data, it almost feels like a contradiction.
How so?
Well, we just established that the PD8's microenvironment neutralizes the CD8 T cells and the CDC1s. The main RE is dead in the water.
Right.
So if standard immunity requires those cells, how did the triple therapy eliminate the tumor? Who is doing the killing?
This is the critical pivot of the entire study. The authors didn't just observe the tumor shrinking, they ran cell-type specific depletion experiments.
Meaning they systematically removed certain immune cells to see if the tumor still died.
Exactly. And the smoking gun was their use of BATF3 knockout mice.
Okay, BATF3 knockout models. Let's explain that for the listeners.
Yep.
By using that specific knockout, you are running an immune system completely devoid of CDC1 cells, right?
Precisely. The spotters literally do not exist in this mouse model.
What happened?
The therapy still worked. The tumors were still completely eliminated.
Oh, wow.
Yeah. By achieving tumor clearance in a model completely lacking CDC1s, and through further depletions showing it was independent of CD8 cells too, they proved something massive.
That the triple therapy wasn't just somehow rescuing the suppressed frontline cells.
Right. It was utilizing an entirely separate, redundant biological pathway.
That is wild. It's like finding out a modern building has a completely mechanical hidden backup generator that kicks in when the digital grid fails.
I really like that analogy. The triple therapy bypassed the CD8 and CDC1 blockade entirely. Instead, the researchers found that the new heroes of this story are CD4 positive T cells and CDC2s.
The type 2 conventional dendritic cells.
Exactly. What's fascinating here is the shift in cellular roles.
Break that down for us, because CD4 cells aren't usually the killers, are they?
No. Historically, CD4 positive T cells are viewed as the supporting cast. They're literally categorized as helper T cells.
Right, they help out.
Yeah, they were traditionally thought to primarily assist the CDC1s in priming the CD8 infantry, or to help B cells produce antibodies. They are the logistics and supply lines.
But under the conditions of this triple therapy, what happens?
The CD4 T cells step out of that helper role and become the primary anti-cancer effector cells. They execute the tumor elimination themselves.
Wait, they just suddenly become the frontline infantry?
Yes, but they don't do it magically. The paper outlines 3 absolute molecular requirements for this new CDC2 to CD4 axis to function.
Okay, let's detail those, because this biological nuance is crucial for you guys listening. What's requirement number 1?
First, the CDC2s must express MHC2.
Major histocompatibility complex class 2.
Right. It's essentially the molecular display case they use to present the tumor antigens to the CD4 cells.
Okay, so the CDC2s need the display case. What's second?
Second, the CD4 cells require a transcription factor called T-bet.
T-bet, what does that do?
That T-bet expression is what drives the CD4 cells to differentiate into a highly aggressive Th1 subtype. Without T-bet, they just don't mature into the specific effector state needed to fight the cancer.
Got it. And the third requirement?
The third is the actual mechanism of cytotoxicity, the weapon they use. The activated CD4 T cells must produce a specific cytokine called IFN-γ or interferon-γ.
No way. I want to isolate that last point, because the paper makes a massive point of emphasizing IFN-γ over other cytokines, right?
They absolutely do.
Because usually, when we look at CD4 effector functions, we expect a cocktail of cytokines, notably TNF tumor necrosis factor, to play a heavy role in killing the cells.
Usually, yes. But the researchers found TNF was totally dispensable here.
Really? It didn't matter at all?
Not at all. It had to be IFN-γ doing the heavy lifting. That's a vital distinction. When they blocked IFN-γ, the triple therapy completely failed and the tumors grew.
But when they blocked TNF,
The therapy continued to work perfectly. The tumor clearance is specifically and singularly driven by the IFN-γ, secreted by these Th1 polarized CD4 T cells.
that completely redefines our understanding of CD4 cytotoxic capability in the tumor microenvironment.
It really does.
Okay, this is the perfect spot for our second 5-minute checkpoint to synthesize this massive shift before we move into the mechanics.
Go for it.
All right, bullet 1. The triple therapy, which is the systemic ICB plus a local STING agonist, destroys PDA tumors.
Bullet 2. It completely bypasses the jammed front door, meaning it doesn't need the blocked CD8 positive T cells at all.
And bullet 3. It unlocks a brand new backup generator pathway, the CDC2 and CD4 positive T cell axis, driven specifically by IFN-γ.
Exactly. That perfectly captures the what. But the true elegance of this paper lies in how they proved the how.
Right. How does this relay race from the localized tumor to the systemic immune response actually happen inside the body?
Because you don't just spontaneously generate an army of tumor-specific Th1 CD4 cells out of nowhere.
No, they have to be primed. They need to be handed the biological mugshot of the tumor and given the order to attack.
Precisely. And the authors designed 3 brilliant experiments to map this sequence out. Let's walk through them.
Okay. Starting with what I like to call the Z-screen tracker experiment. They needed to prove that the CDC2s were actually the ones picking up the tumor antigens in the first place.
Right. To visualize this, they utilized a genetically modified PDS tumor line that expressed Z-screen.
Which is a bright fluorescent green protein, right?
Yeah. Exactly. So the tumor is glowing green. And by injecting the STING agonist into these glowing tumors, they could track exactly which immune cells were ingesting the fluorescent tumor antigens.
They just looked for the green cells.
Yes. And when they analyzed the cellular infiltrate, it wasn't the CDC1s, the macrophages, or the monocytes carrying the antigen.
It was the CDC2s.
Overwhelmingly the CDC2s. The cells containing the Z-screen, the mugshot as you called it, were the CDC2s.
That's brilliant. And crucially, those CDC2s didn't just stay parked inside the tumor microenvironment once they had the mugshot.
No, they were migrating. Which brings us to the second major experiment, the FTY720 blockade.
Okay, explain FTY720, because the hypothesis here is that the CDC2s pack their bags with the tumor antigen, and then they travel to the tumor-draining lymph nodes, the TDLNs, to present that antigen to the naive CD4 T cell.
Right, they have to go to the command center to brief the troops. To prove this migration was necessary for systemic immunity, they used FTY720, which is a sphingosine 1-phosphate receptor antagonist.
A mouthful. What does it actually do?
Mechanistically, it induces the internalization of S1P receptors on lymphocytes. But practically speaking, it traps lymphocytes inside the lymph nodes.
Ah, so it locks the exits.
Exactly. The T cells can be primed inside the node, but they cannot leave to circulate through the body.
This experimental design is just beautiful. So they set up a mouse with 2 distinct geographically separated tumors on its body.
Yes, a local one and a distant one.
They inject the STING agonist into only one of those tumors, and they simultaneously administer that FTY720 blockade to lock down all the lymph node exits.
And the results perfectly validate the migration hypothesis.
What happened?
Well, under the FTY720 lockdown, the injected tumor still shrank.
Which makes sense due to local immune activity right there.
Right, but the co-transplanted distant tumor progressed uninhibited. It survived.
Which confirms the entire systemic relay race. It proves the CDC2s acquire the antigen at the primary tumor, they travel to the lymph node command center, and they prime the CD4 T cells.
Yeah.
And crucially, those newly activated CD4 cells must then exit the lymph node and travel systemically through the bloodstream to hunt down metastatic or distant tumors.
Precisely. When you lock the lymph node doors with FTY720, you cut off that systemic supply line, and the distant tumors survive.
It demonstrates that the intratumoral STING injection isn't just creating like a localized chemical burn that kills the tumor.
No, not at all. It is generating true systemic full-body anti-tumor immunity via this new CDC2 to CD4 axis.
Which is incredible.
Yeah.
But now we have to address the massive biological curveball in the data, the third experiment.
Ah, yes. The IFN-γ knockout mystery.
I have to admit, as I was reading the paper and tracking the logic, this is where the predictable path completely vanished for me.
It's definitely the twist in the story.
Because we established earlier that the STING pathway acts as a fire alarm, right? And the signature defining downstream effect of STING activation is the massive production of type I interferon.
That is textbook immunology. STING equals type I interferon.
So the logical assumption is that type I interferon is the specific signal that wakes up the CDC2s.
It is the most robust, well-documented assumption you could possibly make. STING activates IRF3, which drives type I interferon, which then binds to the IFN-γ receptor on dendritic cells to activate them.
So the researchers naturally tested this.
Yes, they ran the triple therapy in IFN-γ knockout mice.
Meaning these mice completely lack the receptor for type I interferon.
Right. Functionally, their immune cells are completely deaf to STING's primary alarm signal.
So if the type I interferon hypothesis is correct, the therapy should fail entirely in these knockout mice. The CD2s shouldn't wake up and the tumor should grow.
That would be the logical outcome, but that's not what happened.
Not at all, right?
No. In the IFN-γ knockout mice, the therapy completely maintained its efficacy.
Wait, really?
Yeah. The CDC2s activated perfectly, they migrated to the tumor-draining lymph nodes, they primed the CD4 T cells, and the tumors were efficiently eliminated.
That is wild. It completely upends the expected mechanism. I mean, if the CDC2s aren't being activated by type I interferon, what on earth is translating the STING agonist injection into CDC2 activation?
That is the unsolved mystery that this paper basically leaves at the doorstep of the scientific field. The specific molecular signals linking the STING pathway to CDC2 activation in this specific context remained totally unmapped.
Wow.
Yeah, I mean, STING can activate other pathways. For instance, it can trigger NF-κ, which drives pro-inflammatory cytokines completely independently of type I interferon.
Oh, I see.
It could be some uncharacterized chemokine gradient or perhaps a localized stromal interaction we just haven't identified yet. But the canonical type I interferon pathway, it's officially ruled out here.
I find that so incredibly exciting. It's a perfect reminder that biology isn't a closed book. You run an elegant sequential experiment, expecting to just confirm the standard textbook pathway, and instead you uncover a completely hidden communication channel.
It really highlights the sheer complexity of the tumor microenvironment. We are constantly discovering that these immune cells have alternative, hidden communication networks.
And mapping that non-canonical STING to CDC2 pathway is undoubtedly going to be the subject of intense focus over the next several years.
Oh, absolutely.
Okay.
It's the next frontier for this specific research.
Okay, let's hit our final 5-minute checkpoint to lock in this relay race for you guys listening.
Let's do it.
Bullet 1. The CDC2s basically eat the tumor antigens, they take the mugshot, and they travel to the lymph nodes.
Bullet 2. Inside those lymph nodes, they prime the CD4 positive T cells, which then enter the bloodstream to hunt down distant tumors systemically.
And bullet 3. The STING agonist activates these CDC2s, but surprisingly, it does not do it to the expected type I interferon pathway. It's a total mystery.
That is the complete mechanical picture presented by Qureshi and colleagues.
Awesome. So what does this all mean for you listening right now? We spent a lot of time discussing murine models, orthotopic tumors, and gene knockouts.
Right, mouse studies.
But how does this functional redundancy actually translate to human oncology, specifically for patients battling pancreatic cancer today?
It translates directly and very promisingly. The authors didn't just stop at the mice. They explicitly bridged their findings to human clinical data.
What did they find in the human data?
They analyzed tissue samples from both treatment-naive and chemotherapy-treated human PDAC patients, and they found that these specific populations of CDC2s and CE4 positive T cells are not just theoretical.
They're actually there.
They are physically present within the human pancreatic tumor microenvironment.
Oh, wow. So the cells are already inside the fortress. They just haven't been given the right activation signals to break out of their suppression.
Exactly. The CDC2 to CD4 T cell axis is an accessible, viable therapeutic target in human patients right now. If we connect this to the bigger picture, the concept of functional redundancy is paramount here.
Functional redundancy. Explain why that's the big takeaway.
Because the realization that our immune system possesses a fully capable backup generator using CDC2s and CD4s when the primary CDC1 and CD8 engine is broken could be the master key to overcoming immunotherapy resistance.
And this doesn't just apply to PD8, does it?
No, not at all. This principle of unlocking redundant axes could revolutionize treatments for multiple types of highly fibrotic, immune-evasive malignancies.
Like certain colorectal or ovarian cancers, maybe.
Exactly. It fundamentally expands our entire therapeutic arsenal.
It's so hopeful. But as we wrap up this deep dive, the source text leaves us with a few highly provocative questions to chew on, and I want to pose them directly to you, the listener, as you think about the implications of this research.
There are definitely some lingering questions.
Yeah, first regarding the actual killing mechanism. We know the Th1 polarized CD4 T cells require IFN-γ to achieve PD8 control.
Yeah.
But how exactly does that IFN-γ destroy the tumor?
That's a huge question. Is it inducing direct cytotoxicity and apoptosis in the tumor cells themselves?
Right. Or, as some recent melanoma studies have suggested, does the IFN-γ actually act on the tumor vasculature? Like, does it choke off the blood vessels and starve the tumor through induced ischemia?
And that distinction is critical because if the mechanism is a vascular regression, it opens up entirely new avenues for combination therapies.
Like using anti-angiogenic drugs to amplify the effect.
Exactly.
Such a cool thought. And the second major question revolves around clinical application. We know we need to pull the innate immune fire alarm to wake up these CDC2s.
Right, and in this study, they used an intratumoral STING agonist.
But delivering STING agonists safely in humans is currently a massive clinical hurdle, isn't it?
It is, due to the risk of severe systemic toxicity if the drug leaks out of the tumor bed into the rest of the body.
So if STING is too volatile right now, to what extent can other innate immune triggers accomplish the exact same CDC2 activation safely?
That's the million-dollar question. Could we use localized TLR agonists or maybe newly engineered cytokine prodrugs?
Right. It's the hurdle between the laboratory and the clinic. The mechanism is proven.
Yeah.
Now the pharmacology basically has to catch up.
Precisely.
Well, it leaves me with this final overarching thought for you all. If our immune system possesses a completely separate, highly lethal hidden gear, this CD4 axis for eradicating cancer when the primary CD8 gear is broken, what other redundant defense systems are just hiding in plain sight?
It makes you wonder.
Think back to our murky, fog-filled fortress from the very beginning. For decades, we've assumed that if our frontline infantry couldn't breach the walls, the battle was lost.
But maybe the problem isn't that the armor is impenetrable.
Right. Maybe we've just been hyper-focused on the wrong battalion, completely ignoring the heavy artillery waiting quietly in the reserves.
The therapeutic tools are already inside the tumor. We just finally have the blueprint to activate them.
Thank you so much for joining us on this deep dive into the hidden mechanics of immune redundancy. Keep questioning the established pathways and keep looking for those hidden gears. We'll talk to you next time.